other speakers, I won't thank the organizers because I am an organizer. Instead, I will thank the speakers and the audience for giving us the privilege of having you here, listening to our research and collaborative exercises in bringing leading scientific edge concepts to society and to classrooms. And Brian laid out a beautiful foundation for my presentation, and he focused on the host side of the equation. But as you will see from my presentation, there are two sides to this problem, the host and the pathogen side. What I want to do is first impress upon you the gravity of the situation that we're facing with infectious disease, especially with tuberculosis. And as Brian has already pointed out, nearly a third of the world's population is estimated to be latently infected with this pathogen. And there's about 10 million new infections every year. That's about close to 50,000 new infections every year. And then you have 1.8 million deaths. Now, these numbers can numb you after some time because they're just numbers. But it is worth putting that into perspective. There's a death every 20 seconds. So by the time the symposium is done, 7,500 people who are alive would be dead by tomorrow afternoon. And that's equivalent to almost three cities the size of Seattle being wiped out every year. And that's the gravity of the situation we're facing. And what I'm going to tell you is it's getting worse. Just on Saturday, on the front page of the New York Times, there was this article that impressed upon us this problem across all infectious diseases, that all the antimicrobials that we have been using are becoming ineffective. And there's pathogens that are coming up who are not responding to these therapies that have worked for ages. And so the problem is going to get worse. And if you look in the case of TB specifically, what you see is there are strains that are arising across different continents that are multi-drug or extremely drug resistant, meaning they're not responding to most of the frontline therapies. So new therapies are being released, but the pathogen is figuring out a way to escape any therapy that we're throwing at it. There's a number of reasons for this, including compliance issues with how people may or may not stay with the full drug regimen. But there are other issues as well that I would like to discuss today about how the pathogen can escape therapy. Now, this is not a problem that is just restricted to tuberculosis. If you look at any infectious disease, across all infectious diseases, it's estimated that there'll be about 10 million deaths by 2050, and that would be an annual burden of fatalities. Just from AMR, just from antimicrobial resistance. It'll dwarf deaths by all other major leading causes. And TB will be one of the major drivers of this, is because 1 in 4 deaths currently that occur because of infectious diseases are because of tuberculosis. So we need to understand how we can tackle this situation. And if you look at TB, the current drug regimen, you already have about 6 months worth of antibiotic treatment that a patient needs to take for susceptible strains. And if there's a resistant strain, can you imagine how many pills a patient has to take? Just think of a big number. And then the real number hopefully will shock you. On average, up to 14,600 pills have to be taken by a patient who has drug-resistant tuberculosis. And these drugs have got extreme side effects. So the quality of life of these patients is dismal. Many of them are paralyzed, they're unable to walk, they're bedridden, so they cannot work. So the burden of the disease is even greater, and this treatment can go on for up to 2 years sometimes. So we're facing a huge crisis, and we feel that this is a problem that is inflicting Africa and India. Now, as the climate is changing, as people are moving around the globe, a lot of the tropical diseases are not going to stay confined. They're going to spread for a variety of reasons, human migration included as one of them. The diseases are going to spread, and we're going to get it here in the United States as well. So this out of sight, out of mind is not going to work for too long. We really have to take action right now. And what we have to think of is we have to create a sense of urgency. We're running out of antibiotics. And now popular media are picking it up. Everyone is beginning to notice it, is no matter what drug we come up with, the pathogens can't find a way out of it. So how can we come up with a drug regimen, one drug or a combination of drugs, that is effective? When I say effective, it means they can kill the pathogen in small doses in a short period of time. So in that surface that you're seeing, the surface has to be pushed down so that you have killing with small amounts of drug over a short period of time. That's the holy grail. But even then, if you come up with such a drug, the pathogen can find a way out. And it would be good to understand, what are the underlying phenomena that we have to think about when we design these therapies. Just kill has not worked. There's something else we need to think about. So let's spend some time thinking about it. So when we think of resistance, what we are referring to is a large amount of drug required to kill the pathogen, whereas a susceptible strain requires a smaller amount. But if you look at the susceptible strain, it has some underlying, what one might think of as epiphenomena, which is they can tolerate killing. So they can tolerate killing by triggering some networks or pathways without any mutation, where they can exert some phenotypic change that can prevent killing. So that is called tolerance. Another way they can tolerate killing is by hedging their bets on a small subpopulation of bacteria. So again, genomically they are identical, but phenotypically there's a small fraction of the population that resides in a tolerant state. They call the persister-like state. And so when you kill off all the susceptible cells, there's a small number of cells that remain alive, and they're difficult to kill. So we have to understand there's not one mechanism, but there's many different kinds of mechanisms that interplay in complex ways. And the complex ways are contextual, meaning the context of the host, the context of the local microenvironments where the pathogen resides, and so on. So we need to understand these epiphenomena and the underlying mechanisms in a contextual way. So a primer on how TB infects humans might be worthwhile to think about how complex it is and what kinds of context we have to now consider. So the typical cycle is an infected individual will spread the disease by coughing, and the aerosol containing the pathogens will be then inhaled by other susceptible individuals. They get infected, and as Brian pointed out, some of them progress to disease. The others may go into this pool of latent infections where they act as reservoirs of the bacteria. They can get reinfected, and they can also disseminate the bacteria when they get reactivated, when the immune system goes down, especially. And so on, the cycle continues. And this is what TB has evolved to do since it's existed, and it's been able to understand the host inside out, literally. And it has devised all these mechanisms to evade the host attempts to kill it. So let's look a little deeper. So when the pathogen infects the host, as again Brian has pointed out already, so I won't belabor the point, it is engulfed by macrophages, and the macrophage actively tries to kill it by sending in phagosomes or lysosomes to merge with the phagosomes and lower the pH. And you'll see, or I'll mention later on, that TB has evolved pH-sensitive programs that can then be elicited to prevent killing by the acidified phagosomes. Furthermore, it then uses the immune attack as a way to build a house for itself, which is called a granuloma. So all the dendritic cells, the T cells, B cells, macrophages come and try to kill the infected, the pathogen within infected cells. And over time, the pathogen is able to take advantage of that, change its physiology, kill these cells, and proliferate. And it goes through these sequential stages, and depending on the individual, they can stay in a latent form for an extended period of time, but when it gets active, it goes into this gaseous granuloma, and then it is disseminated, and the infection cycle continues. And you can see the complexity of the microenvironment and the spatiotemporal dynamics associated with the infection cycle. These are all the things we need to consider to then figure out which mechanisms is TB then deploying to deal with the host attempt to kill it. And there's different kinds of challenges you can think of: the host immune attack, the hypoxic environments within the host, the low pH, nutrient depletion, and so on and so forth. And you can also have us trying to kill it with drugs. And it's seen this over and over again. So it has come up with this toolbox to escape all of these attempts by us to kill it. And we have to figure out what are the mechanisms by which it hedges bets on these persister populations. Turns out there's not one, but there's at least 70 or more mechanisms that can generate these persister cells. And they can be activated stochastically, and I'll show you results on that in a minute. It can remodel its cell wall and make it impenetrable. It is already impenetrable, but it can further remodel when it infects the host cell. What are the networks that control it? What are the chemical properties of the cell walls? And where are the vulnerabilities in the networks in the cell wall that we can exploit? And so on and so forth. So what I'll tell you is work done by a very talented group of people in my lab, and I've put most of them up here, but I've highlighted the people who contributed with red borders. And I want to especially call out Eliza Peterson in my lab, who's really heading the TB work. She's here in the audience, and I would encourage you to go talk to her if you want to know more about some of the technical details. And there's a whole bunch of other people, and I'll put names to these faces as I go along in my presentation. So in interest of time, I don't have enough leeway to tell you about all the technologies we've developed, but I'll touch upon few. So there's one technology that Vivek developed specifically to get at the persister population. Now, the traditional way people have looked at persister populations have been by killing off all the susceptible bacteria with antimicrobials and then going after the few cells that are remaining behind. Now, the challenge with that approach is the treatment with antibiotics itself can elicit some protective response by the pathogen. So you can't differentiate the stochastic behaviors from the antibiotic-induced behaviors. So what Vivek did was he developed this technology to take advantage of a physiologic state of the persister cells, which is called translational dormancy, meaning they can transcribe their genes, but they cannot translate them into proteins. So he then developed this reporter system, which will allow them, the cells across the population, to transcribe this, but only the persister cells are not able to translate the transcripts to make the fluorescent reporter. So when you sort them, the dim population that is alive, that is not fluorescing, is the pre-existing persister population. And I'll tell you more about that in a little bit. So that gets us to the bet-hedging strategies of looking at population structure. Now, in parallel, we've developed reactor systems that allows us to then control the growth conditions. And as you heard from Brian, it's very difficult to work in the BSL-3. So we had to take some of the age-old reactor technologies and then modify them so that they could be used in the BSL-3. So we could put remote sensors and track the flow of oxygen within the container where the pathogen was growing remotely from outside of the BSL-3 and go in time to collect samples without perturbing the cultures. And this was done by Abrar and by Eliza in collaboration with each other. Further, this is all work that is being done in flasks and in reactors. The true host context is paramount. So Eliza developed this technology called PathSeq, which allows you to now enrich the pathogen component of the RNA, which can be less than a 200th of the total RNA population within infected cells, and sometimes even in lower amounts. And you can then quantify the transcriptome of the cells within host cells. Okay. Now, from each of these experiments with persister cells, with growing them under very controlled environments or from host cells, we get a snapshot of the transcriptome. What we really want to do is then uncover the gene networks that are active and responsible for generating those states. So what we've done is we've gone across about 30 different studies from 53 laboratories, taken all the data sets that were generated in experiments that probe the response of MTB to different environments, including host infection, collected a compendium of 2,300 different transcriptomes and generated a network that was predictive of all of its responses to different environmental conditions. We can take that network and we can take the transcriptomes that are developed and that are collected from the different contexts I described, and we can then discern the active transcriptional programs of TB within infected cells in specific environmental contexts. So we can then not just look at all the possible interactions, but which interactions are truly active and responsible for the phenotype we're observing. And a whole bunch of people have contributed to this effort, and I mentioned the leading investigators in this effort. So with these technologies, we can now begin to uncover the complex biology of TB, and I'll give you some highlights of what we've uncovered. So what Vivek has discovered is that when you grow a clonal isolate of TB in a flask, it can stochastically form persister-like cells at a very low frequency, as low as 0.5% or lower. And so every time a cell divides, it has a 99.5% chance of giving a daughter cell that is also an actively dividing cell, and a 0.05% or 0.5% chance of giving a persister cell. Okay? And so if you try to kill this population, that persister population survives because he showed that they're at least 4-fold or higher tolerant to killing by frontline antibiotics like isoniazid and rifampicin. Okay. Then he took individual cells from these susceptible population and the tolerant persister population and applied single-cell transcriptomics to figure out which of the different mechanisms for generating persisters were active. And then based on that analysis, he found that there were overlapping subpopulations of cells, and there was multiple different kinds of mechanisms. He identified at least 3 distinct mechanisms in the experiment that he did, which told us that there is this phenomenon where most of the cells lie in this preferred state where they're actively dividing and they're susceptible. But at low frequencies, some of these mechanisms I mentioned, the toxin, the antitoxin mechanism or the relase protein mechanism get activated, and some cells jump out of this preferred well and they go into this low probability wells where they reside in a tolerant state. And they can jump in and out of these wells. So anytime you try to kill it, so no matter what antibiotic we come up with, these cells have a way to escape it, even not knowing what the antibiotic is going to look like. And the situation gets even more complex. So when the pathogen encounters a host environment, such as nutrient starvation type environment, the probabilities of switching shifts. And now every time a cell divides, there's about an 83.5 or 91.5% chance that it'll give you a persister cell, and a lower frequency that the persister cell will divide and give you back the susceptible cell. The population almost entirely shifts to a persister-like state. So even before we start the war with the pathogen, it has put up its armor. Okay? So all of the screening we do for drugs is typically with the susceptible population. What we're trying to kill is the tolerant population, the persister-like cells. So we are dealing with apples and oranges in terms of the molecules we develop and what we're trying to treat. So the question is, how do we ever get around this? If there's 70 mechanisms in combination, that's an unprecedented amount of ways it can generate persister cells. What Vivek then figured out was using some probes for oxidative metabolism is that the cells, regardless of what mechanisms generate them and whether it's nutrient starvation or not, they converge to a low oxygen metabolism state. And he hypothesized that if you were to activate the oxidative metabolism, you could kill these cells. And he managed to do that with cysteine. And he showed that when you add cysteine, the killing effect of antibiotics is potentiated by 2 to 3 orders of magnitude. So if we have a way of activating cells, which is a highly controversial thing to do, especially if there's a latent infection, but if you're able to activate cells into a more actively growing state with a small molecule that triggers oxidative metabolism, you can kill TB. Okay? Furthermore, now this is one of the contexts in terms of the persister subpopulations in nutrient starvation, but upon encountering the host, the pathogen also has to deal with a hypoxia type environment. And that has been a proxy for understanding how the bacterium goes from a dividing state to a dormant state within latent granulomas. And the way people have done that traditionally has been growing a culture and then stopping shaking, and the cells suck up all the oxygen and it shifts to an anoxic state. So it happens almost instantaneously. So what people had understood from that is that there's about 300 genes that are associated with transitioning from a normoxic environment to a hypoxic environment, which doesn't seem to be entirely the complete picture. And our conjecture was it's because the experiment design was not controlled well enough, and there wasn't enough sampling that allowed us to look at the transition of the pathogen into and out of hypoxia. So we used the reactor I described earlier, and in slow motion, we then observed how the pathogen changed its transcriptome as it went from a normoxic environment to a hypoxic environment and out of hypoxia. And we were able to collect a series of time points because we had slowed down the transition. And we saw that instead of 2 states, there are actually 6 substates that exist that covers about 3,000 genes, or about 60% of the genome of the pathogen. So you could see how it transitioned from this normoxic state through these different states and out of hypoxia back into a resuscitation stage. And because we were able to collect the transcriptomes using the network biology I described in one of my earlier slides, we were able to then map the active circuits within each of these 6 states. And we were able to map which were the regulators that were responsible for controlling the physiology in that state. And furthermore, we were able to then figure out across the states there was some kind of coordination where a preceding state was then controlling the upcoming state and also the state that occurred before it. And so this enormous coordination allowed the pathogen to do a number of interesting things. And looking at the circuit topologies, I'll just mention a few things that are really noteworthy here. There was robustness in the response of the pathogen, so it did not just shift to a hypoxic state upon sensing the smallest drop of oxygen, and it was able to do that through what we think is a fold change detector, which is a mechanism by which it was able to wait until there was a sustained drop in oxygen level to then trigger this transition. Once it committed, it then induced this time delay element. The time delay element allowed it, so once it committed, it took 40 hours before it went to this state here, to stage 2, which is the hypoxia state in which you would find pathogens in latent infection. It's a dormancy-like state. And what we found was the 40 hours was a time frame that then elicited a few additional substates that prepared the pathogen for the long haul. So it remodeled its cell wall, it changed the internal physiology to be more anoxic-like, and it did a bunch of other things, and I'll mention one more really interesting thing in a minute. And it resided in this state, and when there were favorable conditions, it was able to reactivate, but reactivate in a way that it was anticipating that when the host is around, it can come out like gangbusters to infect the host. So here's the interesting thing that we call adaptive prediction or anticipation. The pathogen, when it sensed sustained drop in oxygen over the 40 hours, had prepared itself to then prepare these proteins called PPE proteins. These proteins have been also associated with generating antigenic variability in the pathogen when it infects the host. The interesting thing is these proteins are exported by the type 7 secretion system that is not turned on until when there's favorable environments in the next state. So the pathogen has prepared itself to infect the host when conditions become favorable by making the type 7 secretion system, which then pokes a hole into the host cell and then releases all of these PPE proteins. And in parallel, it can also release proteases and it activates transposases, which suggests that it's generating antigenic variability and genomic variability to create the possibility of escaping host attempts to kill it. So all of this complexity is encoded in this program, which we were able to uncover because we were able to slow down the transition of the pathogen from normoxic conditions to hypoxic conditions. So all of this is still in a in vitro system, inside a flask, inside a reactor. As I mentioned, host context is paramount, and ELISA has developed a technology to look at the pathogen state within host cells using the technology called PathSeq. Using these data and data from bone marrow-derived macrophages of infected mice and alveolar macrophages, she looked at the different states that TB resides in within the infected host and figured out that over time, as soon as it infects the host, it triggers a program that controls the fatty acid metabolism enzymes to change its cell wall. So under normal conditions, when it's actively growing outside of the host cells, it makes nominal amounts of these 2 enzyme RNAs, which allow it to make new cell wall as it divides. But when it infects the host, it then de-represses and makes a lot of these transcripts that completely transforms the cell wall. And then when it goes into a dormant state, it can then shut down mycolic acid biosynthesis. And this is work we did in collaboration with Apurva Bhatt and Rebecca from University of Birmingham, and Mario helped with a lot of the network analysis. I'm giving you this example to highlight how knowledge of what programs get activated can allow rational strategies to disrupt the pathogen activity. So ELISA predicted that if you target MAD-R, you should render the pathogen susceptible to host attack, and it should be lethal. So she targeted it using an overexpression system, which is induced by tetracycline. So the regulator is expressed from a promoter controlled by tetracycline. And she showed both for TB, I'm showing you just a TB data, but also for a related strain, BCG, which is the vaccine strain, and also for a distant relative, smegmatis, that disrupting this regulator can lower the susceptibility of the pathogen by 2 to 3 orders of magnitude. Okay, so the ability to map intracellular state has opened up new ways we can drug the pathogen. Now, all of this is even before we added any antimicrobial agents. So when you add antimicrobial agents, what ELISA and Shui from David Sherman's lab figured out was the pathogen is able to use its evolutionary memory to trigger networks that can then tolerate killing effect of the antimicrobial agent. And the one that she looked at was bedaquiline, which is the first drug in 40 years to be approved by the FDA. And that drug has a delayed killing effect. And what Shui and ELISA figured out was it kills the pathogen by inhibiting the ATP synthase, but that triggers what the pathogen remembers from its evolutionary history as a nutritional stress-like state. And it has evolved these networks controlled by 2 regulators that control about 70 genes that can then express efflux pumps, cell wall remodeling, detoxification, and so on, that goes on and then inhibits the effect of the bedaquiline drug. And she showed that if you were to disrupt these 2 regulators, you can potentiate killing of the pathogen by bedaquiline, and you can get the effect of the knockout of one of these regulators, RV880, with a small molecule called pretominid, and you can combine pretominid and bedaquiline to get a synergistic effect. What was interesting was once she figured it out, we realized that pretominid and bedaquiline are in clinical trials, and they don't understand the mechanism of synergy. And we showed that if you were to overexpress RV880, you can get rid of the synergy mechanism. So we have figured out the mode of action of synergy between 2 different compounds, which was found by brute force, which means we're sitting on a gold mine of a number of additional potentiating combinations within our networks here. Finally, after all these attempts, we would still expect the pathogen to escape. So this is work on E. coli done by Mario with help from Deep and Min. And what he's seen is that in tetracycline resistance in E. coli, when the strain becomes resistant, it rewires its whole network. So when it sees the drug, it elicits a completely different response compared to the susceptible host from which it was derived. And he figured out that the network has changed dramatically, and there's new sets of regulators that have become important to support what we call overflow metabolism. Overflow metabolism is a physiologic state you can think of as similar to fermentation, but in the presence of oxygen. So the way tetracycline is tolerated is by generating large amounts of efflux pumps that take up the real estate on the cell membrane, so the pathogen can no longer use oxidative phosphorylation with the electron transport chain. So what it does is it shifts to fermentation-based energy production. So this overflow metabolism is required to support DNA resistance in the resistant strain. So he predicted that if you were to knock out this regulator, we could cause potentiation of tetracycline and make the resistant strain susceptible again. And he showed that. So tetracycline is a bacteriostatic drug, so it takes a really long time for the wild type in small amounts of drug to grow. So this is lag phase of growth. The resistant strain, on the other hand, can tolerate large amounts of drug. And if you remove the RK network, it can make this resistant strain susceptible yet again. So what I've shown you is there's this complex set of spatiotemporal dynamics in how susceptible cells give rise to persister cells. We can think of targeting this type of a spatiotemporal network in a number of different ways. The primary drugs can target the susceptible cells, but we can also now think of ways we can block this stochastic transitioning of persister cells to susceptible cells and back, so they can all become susceptible and then get potentiated for killing by the primary drug. And we can also specifically target the persister cells using triggers of oxidative metabolism like cysteine. But if it escapes to become resistant, there are ways we can then think of targeting the new vulnerabilities that are exposed in the pathogen because it became resistant. Now, there's so many different ways you can combine the drugs, but remember, 14,600 pills, we can't add 6 more drugs to that combination. We really need to think of ways we can rationally formulate combinations that are most effective for the type of infection an individual has. So we're developing strategies for doing laboratory evolution, and I'm just show one of these data sets. And this was actually originally developed in Natalie Balaban's group, where she used it to study evolution of resistance in E. coli. We applied it to tuberculosis, and because of lighting, you can't see it, but we can observe in real time now the growth of these pathogen cells that have been pre-treated with some antibiotic, and we can do it over different time scales, and look at how the cells are able to grow immediately with a resistant mutation, or after a prolonged period of time, they can then begin to grow. So looking at all these properties, we now figured out from sequencing all the different ways resistance can emerge, that there really are 2 principal ways you can classify these pathways of emergence to resistance. One of them would begin with a gain of a resistance mutation early on upon experiencing the antibiotic. That lowers fitness dramatically. A subset of the population can then gain a compensatory mutation and then able to take over the culture. These types of trajectories are less likely to be effective in a natural infection. The most likely route would be this one, where a compensatory mutation that fixes tolerance arises first, and that enhances the fitness of the pathogen in presence of the drug, and that is followed by a resistance mutation. If this turns out to be correct, what we can then do is think of ways we can monitor infected individuals for signatures of trajectories towards resistance. And depending on the type of mutations, we can then formulate drug combinations that are going to block those resistance mechanisms so we can prevent emergence of resistance. So we think of a 4-way combination along with the primary drug that could then effectively kill the pathogen, starting with blocking pre-existing persistence, preventing expansion of persistence, blocking a drug-induced tolerance like the one with bedaquiline, and ultimately reversing resistance like we showed with tetracycline. And to transfer this to the clinic, what we are now doing is now we have to drive advances in diagnostics and drug development and vaccine development. And as a small academic institution, we can't do this all on our own. I don't have time to go into details of how we're driving these collaborations, but we have collaborations within the US across, and these are all active collaborations, all of these different entities. And with Brian, we're just starting a collaboration to look at how we can apply PathSeq to look at the pathogen susceptibility to drugs in different host cell backgrounds. We are working with IDRI and also with Cornell to look at therapies that are targeting the pathogen or the host cells. And a lot of this work is being funded by the Bill and Melinda Gates Foundation and NIAID. We also have collaborations in Canada and Mexico where we're helping smaller labs design vaccine trials and find new regulators of cholesterol metabolism. In Europe, we have partnership with the University of Birmingham, I mentioned that, to look specifically at cell wall remodeling. We have discussions with GSK about drugs that are in the pipeline that we can then use in some of the studies that the Gates Foundation is funding. We are bringing the PathSeq technology to diagnostics with a collaboration in South Africa. And in India, we are helping a group in Indian Institute of Science also understand some RNA editing technologies within the pathogen. So in the last minute, do I have any minutes left or no minutes left?
No minutes, but we have a break after this.
Okay, so very quickly, Jim touched on this, so I'll just quickly mention there's an active effort across ISB to transfer knowledge to society, and we're doing it by spinning out companies, licensing technologies, and also by transferring concepts and practices to classrooms. And this effort is led by Caroline in the Logan Center, along with Bill Lippitt and Jen, and also in my group in the Systems Education Experiences program I founded about 17 years ago by Claudia, who's leading it along with a bunch of other very talented people. And the information is here, but in a nutshell, what we're doing is we're developing curriculum modules by leveraging scientist educator, teacher partnerships, and these modules then get piloted in classrooms, and then we get feedback, and we take the feedback to optimize the modules, and then through teacher training, we then disseminate it broadly. And over the years, as Jim pointed out earlier, this has gone across the globe to over 100 countries and being used by over 2.8 million students. The California State Board of Education adopted some of the modules as official curriculum, and Texas and Washington are likely to follow suit, hopefully. And what we realized as we were developing these curriculum modules is that the usage across the world, India was the second largest user, and coming from India, I realized that you can't use curriculum developed in the US and disseminate it to India. It loses local and contextual relevance, so we are now replicating the whole program in India. And this is a student who is in the program in Hyderabad, in Telangana state. And we are taking modules that were developed here, making it more relevant to problems that the students face in their local neighborhoods. And now we're going to develop new curricula, one of them being on antimicrobial resistance. And doing it with them, it makes more sense than doing it here where we don't have the appropriate context. So in summary, we have, I've shown you how we can go from concepts of understanding these epiphenomena of tolerance and resistance to translation through a global network of partnerships, and ultimately through education we can create awareness and get the whole population engaged in this global problem. So I'll stop there and don't know if there's time to take questions. I can wait.
We probably have time for one question. Otherwise you can catch Nitin during the 10-minute break.
This might be a simple question. So when you have these quiescent or persisters, the quiet states, they're always referred to, and this is oncology too, as basically a single state where the cells go quiescent. How do you know? I don't actually think there's any evidence that it's just one state and there's just one way to go quiescent and one way to come out of it. I would think there would be many ways.
We do know there's many ways to get into a quiescent state. Whether there's many kinds of quiescent state, I don't know yet. I think that's what we're trying to figure out. From the little analysis we've done, and what others have shown, is that there are some common themes to that quiescent state in terms of physiological quiescence and so on. But in terms of the actual mechanistic networks that are active, I don't know yet. But I think it's more likely to be converging on some set of finite number of states. And what we have to look at is what's the common denominator that we can then target.